Hello. First of all, thank you for inviting me. It's very nice to be here. My talk will be a bit of a shift from what we heard all morning, mostly because we're not studying autistic folk, but we are studying genes that are associated with autism. And I'll explain a bit more in detail. But first, I want to introduce the people that actually do the work. This is my lab. And this is in my living room for the Christmas party. Um, so maybe a bit more than a decade ago, researchers got together and tried to understand, is there something that we can learn how autism might arise? And one lead is that there's quite a big genetic component of autism. And so that came from twin studies. The likelihood of being autistic when your twin, your identical twin, is autistic is really high, like somewhere around 90%-ish. And so that indicates that genes are very important to understand. And so people got together and sequenced autistic people and came up with a list of genes. And so what was found is that these genes often have an impact during embryonic development. In particular, a period of time when the neurons, the cells in the brain, find each other and connect with each other. And so my lab tries to understand what happens during this period, and then what happens if we interfere with the genes that were previously found to be associated with autism. Okay, so just to give you a bit more of an idea, so the neurons in the brain are not just all neurons. There's many different types, and these are called cell types. And so here we have one brain region, and this is one cell type that is labeled in green. These are the neurons in mouse's brain. And you can see from when they first appear in the brain to right before birth, a lot of different things are happening. There's, the first neurons arrive here, then they have two layers, then they have just one layer, and then towards the end, they have two layers, but now they're different and they have totally different shape. So how this process happens step by step is what my lab tries to understand. And then what happens if if we interfere with these genes. One of the things that we also try to understand is not just in tissue that we get from the mice, but also try to understand what these cells actually do in the brain. And so we developed a way to watch these neurons in a living mouse embryo. And we can actually just go to the next slide. And I want to show you how that actually looks. So I'm going to show you two time points. One that is early on, and then one that is a bit later. In the first time point, what you see here, I'll explain a bit in a second. So we're looking at the activity of the neurons, and there's three neurons here, and you can see how when they light up, this is a dye that changes its intensity depending on whether the cell is active or not. And so we can watch these neurons, how they're active. And so again, this one is active, then this one is active, this one is active, and this one is active. So we're literally watching when the brain is first active, and we try to understand if we interfere with these genes that are important for autism, does anything change in this moment? Yeah, and let's just go to the next slide. Here is some of the people that are involved in this work. This is Jeremy, Joe, and Maharise, and they're working on different types of genes. Jeremy is looking at what happens if you interfere with the gene P10. Joe is working on two genes that work together, They're called KDM6B and EZH2, and then Moharis is working on CHD8. If you want to learn more about this, you can go to our lab's website or ask me questions. I'll be around for the lunchtime.